Oh, good evening, friends. So we will move to a new topic here, that is biotechnology and its application. So this is what the last section of this 4 days of class. So I thought to cover many chapters. That's why I prepared all the lessons of class 12th. And it's because of what the time, it's not possible to cover all the people all the chapters, but have gone through some important chapter and see the biotechnology and its application. You know what is biotechnology? Mainly that what the recombinant process only. The process of recombination. That means keeping the genes of some human interest in the genome of some other individual. Naturally, it start expressing that foreign character. So that is what the main concept of what this biotechnology, and it is having a lot of application in many fields. So that only we are going to discuss here in detail its application: therapeutic, diagnostic, agriculture, processed food, bioremediation, waste treatment, energy production. And some of the application only we are going to discuss today, and we'll move little fast so that we will able to cover the entire thing. And after covering all the concept related to the application of biotechnology, so I have to go for discussing one question, because yesterday one guy called me and asked the answer for a question, and it's asked in the last first term examination, right? And come to what the biotechnological application in agriculture. So we have mainly three type of agricultural practices. One is what agrochemical based agriculture, that we started after the independence to improve the food production. You know that M.S. Swaminathan only responsible for such type of sudden increase in the food production in India. That is why he is called as what the father of this green revolution. That is what organic, I mean, agrochemical based agriculture. Then second one is organic agriculture. Their chemicals are not, instead organic matter only used to enrich the fertility of the soil to improve the yield. Then another one is what the genetically engineered crop based agriculture, where we use the crop having some modified gene. It makes that plant resistant to disease, resistant to what the abiotic condition, and able to live in wide, having wide adaptability, and without water, within flood, it can survive. So such type of what genetically engineered crop-based agriculture also for a specific function. This is what the chemical-based, agrochemical-based, applying all kinds of poisonous thing in the field to avoid the pest or to increase the fertility by applying the that fertilizers, chemical fertilizers. Of course, it is harmful to the environment. It is not eco-friendly approach. Then it is what the organic one, using the organic fertilizers to improve the fertility, to enrich the fertility of the soil, and it is good to the ecosystem, but it is a costly approach only. And another one is what the GMO. We are using what the genetically modified plants to cultivate in the field, and because of what the genetic modification, it is able to show lot of what the desirable characters. That is what very important. Nowadays in modern agriculture, we are practicing only the GMO type of agriculture, using genetically modified organisms. So best example for such type of agriculture is what the tailor-made plants, and it supply the alternative sources to industries in the form of starch, fuel, and what you can say, the pharmaceuticals. So here, this is what the tailor-made plant. So tailor-made means what the plant growing for a specific purpose, such as what this high yield, disease resistant, or drought resistant, etc. using biotechnological method only, altering the genetic makeup of the individual and make the individual to produce what the desirable characters. And such a tailor-made plan to supply the alternative resources to the industries in the form of starch, fuel, pharmaceuticals, etc. Because we are cultivating such type of plant for a specific purpose. What modern agriculture in the farmhouse or in the what you can say poly house, right? Then why GMO? Why to go for this GMO genetically modified organism? So to prepare cope with more tolerance to abiotic stress like cold, drought, heat, and salinity, we have to make the plants having more tolerance, so it will not be destroyed even sudden changes happen to the environment because it is having the resistance. And to prepare the pest resistant crop to reduce the use of pesticide. If you use the pesticide for a long run, what happened? It destroy our soil, it destroy the water, destroy the environment. And to reduce the post-harvest loss, to increase the efficiency of mineral usage by the plants to avoid the early exhaustion of soil fertility. If it is absorbing everything from the soil continuously, naturally that fertility of the soil get exhausted. And to enhance the nutritional value of the food like vitamin A enriched rice, we can make varieties of food material with some other combination like what vitamin enriched food. See, this is what the normal rice having only what the starch and little quantity of vitamin A. And this is what the rice that we made biotechnologically having rich quantity of vitamin A. And it is what vitamin enriched rice. This we have developed through this biotechnology only. Then very important topic that is what the BT cotton is what the best resistant plants only. BT. BT means what is a bacteria living in the nature called as what the Bacillus thuringiensis. It is what a free living bacteria present in the nature. And the genome of this bacteria is having a gene. Name of the gene is what the BT gene. BT gene, or it's called as what the cry gene. CRY, cry gene. So this gene present in the genome of this Bacillus thuringiensis produce a protein, and it is called as what the BT toxin. It is called as BT toxin, or we call it as what the cry protein. So cry gene present in the genome of this Bacillus thuringiensis bacteria produce an enzyme or protein in the form of crystal, and we call it as what the BT toxin or cry that protein, cry protein, and it is produced by this bacteria in the form of protoxinal. Protoxin means what? It is not a toxin, but it can be converted into toxin later. Whenever it is getting what an alkaline medium, an appropriate condition, protoxin can be converted into what the toxin, right? And such a type of protein only produced by the Bacillus thuringiensis in their body with the help of a gene called as what the BT toxin gene, or we call it as what the cry gene. Cry gene produce what the cry protein, or BT toxin produced by the BT toxin gene. It is there in the nature. So this gene can be extracted or it can be isolated from the body of this bacteria. Then why can't it insert in the body of what? The plants like a cotton? So what will happen if you are inserting such a gene in the genome of the cotton? As I told you in the morning, this gene or this triplet codon is universal. If you fix it, it will work in the body of a plant. The genome or gene of this bacteria can produce the same protein in the body of a plant, or it can produce in the body of an animal, because it is universal. If you fix what a particular gene present in the plant in the body of animal, naturally animals start producing the plant character. Similarly, we can keep some animal gene in the body of the plant. Naturally, it produces what the animal character. Even we can fix it in the body of a microorganism like bacteria. So bacteria also will produce what the animal character. This is what the way through which we are preparing human insulin in the body of E. coli, right? And such type of Bt toxin gene isolated from the Bacillus thuringiensis bacteria and put it into the genome of the Bt cotton. Naturally, Bt cotton produce such a toxic material in the form of protoxin. That crytoxin produced in the Bt cotton in the form of what? The protoxin only. See, whenever this Bt cotton, that green leaf eaten up by, or the green matter present in the, what you can say, the leaf eaten up by larvae of an insect burst. So naturally, this protoxin, it is in the form of crystal. It will enter into the body of its digestive system. And whenever this protoxin in the form of crystal moving through the digestive system, whenever it reaches to the midgut, midgut is what? Alkaline in nature. So what happened? This Bt protoxin will solubilize it, and this will be converted into what? The toxin, protoxin converted into toxin. And this toxic material attack to the membranous epithelial lining of this intestine and destroy it, so that this larva is not able to survive. This is what the way through which the Bt cotton is getting resistant against such type of bollworms. So go through it. There is a bacteria in the nature called the Bacillus thuringiensis, and this bacteria produce what? Anti-insecticidal protein called as what? The Bt toxin, but it produced in their body in the form of what? The protoxin only. And this protein produced in the bacteria, Bt toxin gene, or we call it as what? The cry gene only. And it is what? The cry protein only. And Bt toxin gene can be cloned, can be multiplied without making any change in the chemical nature, and fix it in the cotton plant genome. That this alien DNA. Alien DNA means what? This foreign DNA. Actually, after inserting in the body of the cotton, this gene is not the part of what? The cotton. It is what? A foreign gene only. But after introducing in the gene, its genome, it became the part of the plant. Start expressing its character in the plants and produce what? A toxin in the body of this cotton. It makes the plant resistant against insect without using the chemical insecticide. No need to use any chemical insecticide. And this plant is resistant to such type of insects because they have a protein in the body which is working as what? A toxic material in the intestine of this larvae. So naturally that will die. So no worm will attack this plant. It is getting what? The resistance against this what you can say, insect pest. So let us see how it's work in the body of this larvae. And this Bacillus thuringiensis protein, that is what? The Bt protein or cryprotein, kills some insect, particularly belongs to some order like Lepidopterans. The tobacco bollworms and armyworms belongs to this Lepidopterans only, or its work in the body of the Coleopterans like beetle, or dipterans like flies and mosquitoes. Such a type of that insect will die due to such type of protein in their body. And this thuringiensis produce protein in the form of crystals only during a particular phase of their life growth. But this crystal consists of an insecticidal protein, but it exists in the inactive form. As I told you, it is called as what? The protoxin. But this protoxin can be converted into what? Very toxic material in the alkaline pH. And whenever this larvae ingest this green matter, along with that, the protoxin also will go in their body. So naturally, this protoxin, inactive toxin, converted into an active form in this what? Alkaline medium, and which solubilize this crystal and you start destroying what the epithelial lining. This is what the way through which what this Bt protein or this what the cry protein, a toxic protein, work in the body and make this plant resistant. See, this is what the what you can say that Bacillus thuringiensis, it's what a free living one available in the nature, and you can see the plasmid here, ribosomes are there, and this is what the real DNA, circular DNA in the bacteria. In this DNA only that gene is located called as what the cry gene or toxic gene, Bt toxic gene. And this gene only produce what this Bt toxin in the form of protoxin. And this only we are isolating from the bacteria and fix it inside. See, whenever it's grab this green matter from the leaf, this caterpillar, whenever this caterpillar grab the green matter from this, naturally that Bt toxin, it is there in the form of protoxin only, and this will be taken inside. And this midgut only provide the alkaline medium. So naturally what happened, this will be solubilized in the alkaline medium and converted into what the toxic material that will attack the epithelial lining of its midgut and it destroy. And finally, that caterpillar will die. And the plant is getting what the resistant against this caterpillar. So first this inject that what you can say protein along with the green matter. After entering into their what you can say the digestive system, a dissolution of protein crystal here, and this crystal, you know, converted into what the toxic material here, and these are the toxic material given in what the yellow color, and this what the triangular with the red color toxic material only. And this toxic material bind to the receptors here on the intestinal membrane, and what happened, it destroy the cytoplasm of this cell which line the internal lining of the midgut, and finally that this will die because it is not able to digest, it is not able to absorb, and this insect will die due to what this poisonous material destroy the internal lining of its midgut. And this specific Bt genes are isolated from the Bacillus thuringiensis only and incorporated into several crop plants such as cotton mainly. You may be heard about different kinds of the Bt products like Bt brinjal, Bt cotton, Bt what you can say binti like. And this gene coded for the toxin is named as what the cry. I told you the cry gene. Cry gene. That gene present in the genome of the Bacillus thuringiensis responsible for producing this protein called as what the cry protein. It's a protoxin only. And several such type of what the cry genes are identified like what cry AZ, cry 2AB, and cry 1AB. These are all to control different kinds of insect only like a cotton bollworm, then corn borer, etc. And this question asked once in the board examination, differentiate this cry from this cry. Whenever this cry written with the capital letter, the C, and it indicates the protein only, that capital C and small ry, normal one, that indicate what the protein only. But if it is written in italics, that CRY, that indicate what the gene, and this gene only responsible for producing the cry protein. And this cry protein is protein only that can be converted into what a toxic material in the alkaline medium, because cry protein, remember, it is what a crystalline one. It can dissolve very easily in the alkaline medium, so that this will be converted into what the very active poisonous toxic material. So this is what the techniques to produce what the pest resistant plant against these what you can say insect pest by inserting what you can say the cry gene. How we will insert this cry gene in the plant? By using what a vector called as what the Agrobacterium tumeficient. Always remember there are three vector. Vector means what? The carrier. Three vectors. If you want to keep something in the plant cell, some gene, foreign gene in the plant cell, we have to go for Agrobacterium tumefaciens. If we want to insert a foreign DNA in the cell of an animal, we have to depend what this bacteriophage like vectors, or if you want to insert something in the genome of a bacteria, always we have to go for what the plasmids only. Plasmids, you may be knowing. I told you that plasmid here, this is what the plasmid, right? This is what the plasmid present in what can say the Bacillus thuringiensis. Every bacteria we can see such extra chromosomal circular DNA, and that can also used as what the vector only for biotechnological experiment, right? Then come to another important topic. Biotechnologically, we can make a plant resistance to the pest. Another method only is RNA interference or RNAi. See, normally nematode worm attack the tobacco plant and reduce what the yield production. It is nematode worm infect the root of the tobacco plant. And name of the nematode worm normally attack or infect this crop is what Melodygene ignonica. It infect the root of the tobacco plant and cause great damage in the yield. No strategy was developed to prevent this infection, which was based on the process of RNA interference. I told you RNAi. And this RNAi can inactivate the mRNA produced by the nematode worm in the plant cell. Actually, for what purpose this nematode is entering into the body of the tobacco plant? Is it to destroy it? No. Actually, the nematode worm is entering in the plant to multiply its number. How it's multiply? By producing what its own protein by using the machinery available in the plant body, right? So after infecting this plant by this nematode produce what mRNA, and by using this mRNA produced by this nematode worm, and by utilizing the machinery of the plant, it prepare its what the protein translation that happening in the body, and finally it destroy the crop and reduce the yield production. But here in this technique, in case of plant, you know that there is a particular RNA, we call it as what RNAi. RNAi is there in the crop. RNAi is there in the tobacco plant. Whenever this the nematode worm produce mRNA in the plant cell having RNAi, cannot produce their protein because mRNA produced by this nematode worm will be silenced, cannot go for what the translation process, so that it will not able to multiply in the plant and plant is getting resistance. If it is not possible to multiply the number in the body of a plant, nematode worm never infect this tobacco plant because purpose of the nematode is to multiply. It is not possible if RNAi is there in the plant, that tobacco plant. RNAi present in the tobacco plant prevent the translation of the mRNA produced by the nematode worm, so that protein synthesis will be prevented and nematode will die and it not able to what you can say multiply its number. So this plant is getting what resistant. So through biotechnology, we can make the plant, no need to apply the chemical chemical pesticides here, because plant is already resistant. It never permit this nematode to multiply its number because it is having, every cell of the plant is having what the RNAi that will silence the mRNA produced by the nematode. And the plant is getting what the resistance. So let us see the mechanism through which it is getting what the resistance against the nematode. This is what the nematode worm, and it infect the root of the tobacco plant and the multiplies number by producing its mRNA and do for protein synthesis in the plant and multiply its number. It will not be possible if RNAi is there inside. Then let us see how to prepare such type of RNA interference in the plant cells. There are some specific type of DNA that is called as what the nematode specific DNA. And this nematode specific DNA we can insert in the plant cell by using a vector. What is vector? It's a carrier. Carrier. I told you three type of carrier. Always to carry the foreign DNA in the body of the plant, we use what the Agrobacterium tumeficient only. And here also we are using Agrobacterium tumeficient as a vector. And by using this vector, we can bring this nematode specific DNA in the plant cell. See, after reaching this nematode specific DNA, it is double stranded only. And after reaching in the cell, it get denatured. Denature means double stranded structure, will be converted into single stranded by cutting all the hydrogen bond present in between the strands in the plant cell, and both will go for transcription. You remember, transcription is what the process of formation of RNA only from the DNA. But normally, only one strand acts as what the template, 3' 5' strand only act as a template. And based on that, new RNA will be produced in between. But here, both the strand is acting as a template. So naturally, it produces two RNA. You know, the DNA, both the strands are complementary. And these complementary strands only produce what these new RNA. So naturally, RNA also will be complementary. Because of what the complementary nature of the RNA, these two RNA go for coiling just like what the DNA and making the bond between the nitrogenous base with the hydrogen bond. Adenine bind with the uracil and cytosine with the guanine, and getting what the DNA like what you can say three-dimensional structure, and it is called as what the double-stranded RNA. Remember, we are inserting only the double-stranded DNA by using the vector called Lactobacterium tumeficient in the cell of what this crop. After reaching that, DNA get denatured, and both the strands of the DNA go for producing what its RNA. And RNAs are complementary, naturally go for pairing and get a DNA-like structure, and we call it as what the dsRNA. And this dsRNA again go for the transcription because it's function exactly like what the DNA only double stranded. And here this DS stranded double stranded RNA go for denaturation and separation. But during this transcription, only one strand acts as a template. That 3' 5' strand of the RNA of double stranded RNA work as what the template, and it produce what a single RNA there. And that only we call it as what the RNAi. That RNAi will be there in the cell of this plant. And whenever this nematode worm infect this plant and produce mRNA in their body, immediately this mRNA produced by the nematode will be silenced by the RNAi already present in the cell. And that only we call it as what RNA interference. So because of lack of facility to multiply its number in the cell of crop, or lack of facility to produce protein by using their mRNA in the plant cell, it will die, and plant is getting what the resistance to such an insect. And this topic is very important, and because it is asked many times in the examination, this process to prepare RNA interference in the plant. First, insert the nematode-specific DNA in the plants by using Agrobacterium tumeficient as a vector. This DNA get denatured in the plant cell and go for transcription. And the transcription of this DNA by using both the strand as a template, then so produce two RNA. Both RNAs are complementary, and this complementary RNA going for pairing to form a double-stranded RNA. We call it as what the dsRNA. And this dsRNA again go for transcription by consider only one strand as a template, and it produce what RNAi. It's nothing but the RNA produced by the double-stranded RNA only. Whenever plant is infected with a nematode, and nematodes start preparing its protein in the plants through the process of transcription and translation. First of all, the nematode worm produces RNA to prepare a protein. But immediately after producing mRNA by this nematode worm through the transcription, that will be silenced. So that translation will not possible, no production of protein. So this is the way through which that plant is getting resistant against such a nematode worm. And it is what a silencing of what these mRNA by this RNAi already present in the plant cell only. So nematode worm never multiply in the body. The dsRNA, double stranded RNA, go for transcription and produce what three type of what RNA, single stranded RNA only, like siRNA, small interference RNA, and shRNA, short hairpin RNA, miRNA, microRNA, and these are all collectively called as what the RNA interference. And this RNA interference break the mRNA produced by the nematode in the plant cell, so that there will be no translation, protein synthesis will be affected, and no chance of multiplying the nematode in the body of the crop plant. Nematode will die due to non-production of the protein. This is what the basic principle behind this technique to make a plant resistance to the disease. We call it as what the RNA interference. So this topic is also very important as far as your examination is concerned. Then biotechnological application in the medicines. Maybe heard about so many therapeutic methods called as what the gene therapy is, what the permanent solution for the genetic defects other than medicines. Then molecular diagnosis of disease. We can use what the biotechnology. Then maybe heard about the genetically engineered insulin. So previously, before using this biotechnology to prepare insulin, actually it was very costly that doctors used to collect the insulin produced by the cattle or pig, and that only injected in the body of the diabetic patient, because it is showing some kinds of the chemical similarity with the human being, but not 100% is similar. So naturally what happened, whenever we inject any foreign material in our body, we will show some kinds of allergic response. So that response was there, what you can say, allergic response was there in earlier after injecting the insulin collected from the cow or the pig. But now that is not there, because we are preparing the exact human insulin by using this E. coli biotechnologically, and now we are injecting in the body, and it's commercially available, and it's with a cheap rate only, and never shows any kinds of what such type of allergic response, because it is 100% similar to the human insulin only. So that is what the things mentioned in the text with genetically engineered insulin, gene therapy, and molecular diagnosis. That is what the things we are going to discuss here. First, we will see the genetically engineered insulins, how to make insulin through this genetic engineering. As I told you earlier, insulin for diabetes was extracted from the pancreas of the slaughtered cattle and pig, because it shows some kinds of similarity with the human insulin. Most of the patients showed allergy or other type of reactions to this foreign protein. Of course, at last it is what a foreign material. So naturally, we will show some kinds of allergic response to reject it. And functional insulin consists of two short polypeptide chain. See, we are producing what the insulin hormone having two peptide chain. All of you know, one is chain A and another one is chain B. And both chain A, chain B link together with the two disulfide bridge. And mammals, including human, insulin is produced in the form of non-functional proinsulin only, and proinsulin consists of one extra peptide chain. That is what the C peptide only. So that human being, see, this is what the structure of what the functional insulin. This only reduce the level of glucose in our body, and it consists of what the two peptide chain. One is what the B peptide chain, and it is lengthier than what you can say A peptide chain. 30 amino acids are there. This is what the amino acid. This is one. I told you for every protein, there is an N with the COH and another one with NH2. Because this in phenylalanine, NH2 group is free, and in case of thiosin, in the another end, that COH group is free, right? And this is what the lengthier one having 30 amino acid. You can see the sequence of amino acid present in the B chain of the human insulin. And it is what the A chain, it is what you can say having only 21 amino acids only. From here it starts and it's ended with what the glycine, right? This amino acid is only 21 in the A chain. And the A chain and B chain connected with the two disulfide bond. Between cysteine here, we can find the disulfide bond, and here between cysteine, we can find another disulfide bond. It connects the B peptide with this A peptide. And another disulfide bond is there between the cysteine here, and it connect what the A chain only, because A chain is not like B, it is go for a coiling. So that only making these two cysteine to connect each other with this disulfide bond. Total 3 disulfide bond and 2 disulfide bond connect the B chain to the A chain only. And third one connect these two Vatican cysteine present in the A chain having only 21 amino acid, and it is what the functional. Functional means what? This is what the insulin released by the pancreas to our body and reach everywhere in the body through the bloodstream, and that will reduce the level of glucose in the body. This is what the functional insulin. But we produce insulin in our pancreas not in the form of functional insulin. We produce in the form of proinsulin only. Proinsulin. In proinsulin, there is what an additional polypeptide chain that is called as what the C polypeptide. And this is what the C polypeptide. From here, right, from here, we can find a C-shaped polypeptide. A C-shaped polypeptide chain made with a number of amino acids here, and it is what the C polypeptide, and it connect what the A chain to the B chain, other than what the disulfide. The two disulfide bond is there to connect. Other than that, there is a polypeptide chain which connect the end of the A chain here with the end of what the B chain here. And it is non-functional, but we produce the insulin in the form of proinsulin only in our pancreas. And it is only for the storage purpose. Pancreas is producing insulin and storing in their cells. Whenever we require this insulin, for example, after taking food, level of glucose will suddenly increase. We have to reduce it. So pancreas will release this insulin into the bloodstream, blood only taking to each and every part. But before releasing insulin from the pancreas, they remove the C polypeptide, then only it will be released. So that means proinsulin, inactive insulin, will be converted into active insulin just before it's released from the pancreatic gland. How the proinsulin will be converted into active insulin? Only by removing the C, what you can say, polypeptide chain only, right? And here we will see what the human functional insulin, and this is what the proinsulin only. And same thing you can see here. This is what the proinsulin. This is what the A peptide and B peptide, and A is smaller than the B and connect each other here with the two disulfide bond. And this is what the A polypeptide connected to the B polypeptide with a C-shaped polypeptide chain. And it is called as what the proinsulin. Whenever this C peptide is removed, this proinsulin will be converted into what? A functional insulin. This is only able to reduce the level of glucose in our blood. Actually, pancreas is producing this insulin in the form of proinsulin and store it. But at the time of releasing, it removes the C polypeptide and converted into a functional insulin that only will be released into the blood and taken everywhere. That is what the conversion of proinsulin into what? the active functional insulin only. See, at the time of maturation for its use, C-peptide should be removed, and C-peptide is not present in the matured insulin, functional insulin. This is done in the pancreatic gland itself just before sending it to the bloodstream. The main problem in synthesizing human insulin artificially is to assemble it in a mature form. In mature insulin, only two peptides, A and B. The main problem is what? It's assembly only. And in 1983, the Eli Lilly is what an American company only prepared the human insulin biotechnologically, and they prepared these two DNA sequence to produce polypeptide A and polypeptide B and insert in the genome of E. coli separately. They made artificially this DNA responsible for producing the polypeptide A chain and for producing polypeptide B chain, and they introduced this DNA responsible for polypeptide A and polypeptide B in two different E. coli, and separately this E. coli start producing polypeptide A and B. Then extract it separately from these two E. coli and go for assembling outside and get what you can say the functional insulin only. Artificially, we are preparing this insulin, human insulin, by using E. coli only. And we prepared what the DNA for producing the peptide, B peptide and A peptide. And this DNA we insert into the genome of one E. coli, another DNA insert into the genome of another E. coli and multiply its number. And these two E. coli produce B peptide and A peptide separately and collect it, extract it, purify it, and go for assembling. Assembling means what? Connect these two peptides with this disulfide bond and prepared what? A functional insulin. Now that only available in the market, and the cost is very cheap. One bottle only 100 and some rupees, 110 rupees, I think, and one bottle is enough for a patient to use for 1 month. And no, what you can say, allergic response, very effective and able to reduce the level of glucose without any creating any kinds of problem. So this is what the procedure to prepare the human insulin. Introduce this DNA sequence to produce polypeptide chain A of human insulin in an E. coli by using a plasmid biotechnologically, and introduce another DNA sequence for producing polypeptide B of human insulin in another E. coli bacteria. Then both polypeptide A and B prepared separately into E. coli. Extract this polypeptide chain, then purify these two chain A and B and connect it, combine it, assemble it outside by creating the disulfide bond. So this is what the way through which we are preparing the human insulin. So we will move to another biotechnological use in the field of medicine, gene therapy. Gene therapy means what? This is what the treatment given to the patient having some genetic defects. No medicine we identified so far to what you can say correct the genetic defects. That medicine is there only to pacify the symptoms for temporary purpose. And gene therapy is what? A permanent solution for it. And one example mentioned in the textbook is what? The deficiency of what? The ADA, adenosine deaminase enzyme. We will go through it. So ADA, it's what? The adenosine deaminase enzyme only. This enzyme is actually very much required for the immunity of the individual. Adenosine deaminase enzyme. And it is produced by the lymphocyte called as what? The T lymphocytes only. Adenosine deaminase enzyme is responsible for the immunal response in the body of an individual, and normally this enzyme is produced by the T lymphocyte only. All of you know T lymphocyte is responsible for the cell mediated immunity. It is more important than what you can say B lymphocytes. So it is some individual this adenosine deaminase enzyme will not be produced, because it is producing what, and it is produced by a gene called as what, the ADA gene, ADA, adenosine deaminase enzyme producing gene, ADA gene, and it is defected. This defected gene never produce what, the functional enzyme in the T lymphocyte, so that person will not have the resistant against the diseases. Cell-mediated immunity is not at all there in such an individual with what, such a deficiency with ADA deficiency. So this can be corrected through different means. We can inject this adenosine deaminase enzyme externally. Regularly, we can inject some dose of what, adenosine deaminase enzyme in our body. But it is not the permanent solution. And another one is what, the bone marrow transplantation, because bone marrow only producing what, the what you can say, T lymphocytes, WBC only is produced by the bone marrow. We can because of the defect with such a bone marrow only, it is producing such a defected T lymphocytes. So bone marrow can be what you can say, transplanted. Thing is that is very costly, and a lot of what we have to go for finding a donor to donate that bone marrow, and chance of getting the success is also comparatively very less. So that biotechnological method is what, the permanent and very easy solution for treating such type of problem for the genetic defect due to the deficiency of what, the ADA gene. So this is what the correct genetic defect of an individual. And gene therapy is the collection of methods that allow the correction of the gene defected. Then here genes are inserted into the person's cells and tissues and treat the disease. And the correction of genetic defect involves delivery of a normal gene into the individual. We can correct this one by giving a normal gene into the body of the individual or in the body of the embryo. And that newly introduced gene will take over the functions of what, and compensate the non-functional genes. This is what normally we are doing in the gene. Suppose one gene is not functioning, in the embryonic stage itself, we can introduce what, a functional gene in the body, so that functional gene that introduced in the body take over the function and compensate the non-functional genes. So this is what the permanent solution for such type of disease. And one example mentioned here, it is what, the severe combined immunodeficiency disorder. This is what the name of the disorder, genetic disorder only. SID, we call it as what, SID, SCID, and this also asked many times in the board exam. SCID, SID, that is severe combined immunodeficiency disorder. What is the reason for the disorder? This disorder is due to the malfunctioning of ADA gene. It is not at all producing what, the adenosine deaminase enzyme. And this enzyme is essential for the immune system to function, particularly for the cell-mediated immunity. It is produced by the T lymphocytes only. T lymphocyte is having nucleus with what, the gene in the cell nucleus, and this gene only producing what, this enzyme, and name of the gene produce the adenosine deaminase enzyme is what, the ADA gene. It's having some defects. So naturally, it is not producing that functional adenosine deaminase enzyme. Then how to treat it? As I told you, enzyme replacement therapy is one method. Bone transplantation is what, another method, and biotechnological gene therapy is also another method. But the best method to cure this disease is what, the biotechnological gene therapy, because easy to do it and no side effect and no much expenditure also, right? And here let us see how to do this one. These are what you can say, disadvantages of various therapy. You just go through it. That bone marrow transplantation is very costly process and also required a suitable donor. Enzyme replacement therapy, it is that continuous introduction of such enzyme should be there, and success of this therapy is also slightly meager. and that is not a complete curative type. So biotechnologically based gene therapy is the only possible solution for such a type of defect. It is permanent solution and comparatively cheaper and required very simple procedure only. Can do it very simply. So let us see how to do this biotechnologically based gene therapy for such patients. Simple process only. We will collect that T lymphocyte from such a patient, because the T lymphocyte is not at all producing adenosine deaminase enzyme, because it is having that defected ADA gene, that we have to take it out. Then by using a vector, and here vector is what the retrovirus only. By using this retrovirus vector, we can insert a functional ADA gene into the genome of this what the WBC taken from the patient. Now what happened, that introduced the functional gene is take over the function of the defected gene, that we have to keep it in the culture medium and allow to multiply its number. Then after some days, this what the functional WBC can be taken into the body of what the patient. The same, because it is what nobody can say that allergic response, because this WBC is taken from the individual patient itself, but with the defected ADA gene only. But after replacing what this functional DNA in the genome of that WBC T lymphocyte, it can be taken inside. So what happened, after entering into the body of the individual, it start producing what this enzyme. So with this enzyme, that person is getting what the resistant back, immunological response is getting, so that resistance to the diseases, particularly the cell mediated immunity. This is what genetically engineered lymphocytes. Lymphocyte having a foreign DNA, a functional DNA, and it is what genetically engineered or genetically modified lymphocyte that can perform the normal function of the lymphocyte in the body of the patient. So as it's immortal, after some time what happened, this will because it is having its own lifespan, this, but we have to insert this WBC. It's not very costly. Once in a while we have to give such type of what the modified genetically engineered lymphocyte into the body of the patient. With that, this problem can be cured. So biotechnologically based gene therapy, lymphocyte from the blood of the patients are grown in a culture medium outside. That is what the in vitro. And functional ADA gene is then introduced into the defected lymphocyte of the patient by using a vector called as what the retrovirus. And this DNA will become the part of its genome. And after multiplication of this lymphocyte in the culture medium outside the body, again, it can be taken back to the patient, and these lymphocytes are immortal. It is mortal only. There is a definite lifespan. So patients require periodic injection of such a genetically engineered or modified lymphocyte. But introduction of the functional ADA gene in the early embryonic stage is what the permanent solution for the defect. So this is what the biotechnological way to cure such type of the genetic defects. Right. Then see the diagram. This bacteria is carrying the plasmid with the cloned normal human ADA gene. This is what the cloned DNA ADA gene present in the what you can say functional T lymphocyte that we have to insert into the what you can say the bacteria. In the bacteria, it can be multiplied. And such a type of what the DNA that after multiplication can be incorporated into the virus, because virus is what the actual vector here to transfer the DNA into the human cell. After incorporating this cloned DNA in the virus, retrovirus, this is what the tDNA collected from the sick patient, not having that gene, to produce what that particular enzyme. enzyme is not produced. Adenosine deaminase enzyme is not produced by this T cells of this sick patient. ADA gene is not there. So this ADA gene is incorporated with the genome of this virus, and it is infecting that the T cell and inserting or introducing that foreign DNA into it, and it became the part of what this genome of the WBC, and multiplies, and by keeping it in the culture medium, after multiplying, getting the enough number, it can be taken into the body of the patient. So naturally the patient start producing normally what the adenosine deaminase enzyme. With that, it is able to get the resistant against disease causing microorganism, and immune system will work normally because it is having now a functional T lymphocytes. So this is what the way through which we make a patient all right by using what the functional gene inside. Then another one is what the molecular diagnosis. Molecular diagnosis. To diagnose the disease in the early stage, normally common method is what we will analyze. These are the conventional method. We use the serum, urine, or blood, or so many things we will take from the body of the patient. We will go for a microscopic observation and we will identify. But normally it is not possible to identify such type of diseases in very early stage. Why it is not possible to detect in the very early stage? Because the amount of what the viral genome or the bacterial genome in the blood is very less only. Through normal testing, it is not possible to identify, because in the early infection stage, its content is very low. So how to identify in the early stage? We have to multiply this. So this is what the easiest way to multiply. Maybe heard about the PCR techniques that we are doing outside the body only. We are not using vector. It is what the vectorless method to multiply. Millions of copy we can prepare within a short period of time. Within minute, we will able to make millions of copy of the gene without changing the chemical nature. If one gene of a virus is there in the blood sample, through the PCR technique, we can make millions in a minute. So after multiplying this by using the polymerase chain reaction, of course, we can go for the normal test, so we will able to detect it in the early stage. So this is what polymerase chain reaction is helping for the molecular diagnosis of the disease in the earliest stage. Then maybe heard about the ELISA. I told you, enzyme linked immunosorbent assay. It is what the preliminary test to detect what the AIDS like virus only, or HIV like virus only. And it is more effective to diagnose the disease in the early stage. DNA recombinant technology, polymerase chain reaction, enzyme linked immunosorbent assay like ELISA, etc., are the more effective way to diagnose disease in the very early stage. Because in early stage, the content of what that genome of infected individual like a bacteria or virus will be very, very low. Then PCR methods. You already learned about the PCR in the first chapter. PCR, polymerase chain reaction. So presence of pathogen like bacteria or virus present in the body of the patient in the early stage is very meager and very low. It is not possible to diagnose this. Diagnosis is very difficult in the early stage. So it is not possible to produce the symptoms of the disease. AIDS-like disease show the symptoms only after 5 or 6 years of infection, because the content of what is genome is very less in the early stage in the body of the patient. Pathogens are able to produce the symptoms of the disease only after its multiplication. See, the disease like AIDS produce the symptoms only after 5 to 7 years of infection, depend on the health status. Suppose the person is very healthy, it takes 7 years to show the symptom. If person is very weak, naturally it appears the symptoms of the disease what after 3 or 4 years of infection. This can be detected in the early stage through the polymerase chain reaction. Multiply the number of the genome of that infected individual so that we will able to what you can say detect it in the early stage. And PCR is now routinely used to detect the HIV in the suspected AIDS patient. It also a very powerful technique to identify many other genetic disorders. So PCR is the easiest way to multiply the number of the genome of an infected individual like a bacteria or virus without making any change in the chemical nature, right? And see that detection of gene mutation biotechnologically. So gene mutation can only lead to what the cancer like a serious problems. And this can be detected biotechnologically, detection of gene mutation biotechnologically. So it can be detected in the early stage biotechnology. So select the DNA to be tested to identify the mutation. There are some steps for it. First, we have to, because after seeing the symptom of the disease, we are suspecting this gene is having some kinds of mutation. We have to take such type of the DNA from the patient. And this DNA strand, double stranded and made it was single stranded through the denaturation, break all the hydrogen bond and converted into what the single stranded. Then now we have a single stranded DNA. And by using the DNA as a template, we have to make a complementary strand. But complementary strand, we have to make always with what the radioactive nucleotide only, and we call it as what the probe. That suspected area of the DNA only we are testing biotechnologically to detect the gene mutation. We want to identify whether that gene is mutated or not. So first we will take that particular portion of the DNA and convert this double-stranded DNA into single-stranded one and prepare the complementary strand of the DNA artificially by using what the radioactive probe, and allow this, but you can artificially synthesize the DNA strand with this radioactive probe, hybridize with that single-stranded DNA collected from the patient, and get the double-stranded nature and clone this DNA through PCR technique and detect by using the autoradiography. What is autoradiography? Just I told you, just to keep it, what you can say, on the X-ray sheet, because the probe always emit the light and that can be detected in the X-ray sheets. That will reaching in the X-ray sheet in the form of band. So mutant genes will not appear in the photographic film. Mutant means what? We are that new strand we constructed artificially is having the correct sequence. Suppose that DNA taken from the patient is having some defect, this will not match with what this radioactive probe that we prepared with the correct sequence and go for pairing. If mutation happened in the DNA, that will not make what that particular hydrogen bond. So radioactivity will not be there. So whenever we expose such type of double-stranded DNA under this, what you can say, that X-ray sheet, wherever the mutation happened in the DNA, there we will not get the marking because such area is not getting what? The radioactivity. Radioactivity, that one strand is radioactive. And whenever it is bind with what the another strand taken from the patient, because of the hydrogen bond, what happened that single strand taken from this patient also getting the radioactivity. But if it is not linked properly due to mutation, such area will look blank in the what you can say this photographic film, and this is what the way through which we detect the mutation biotechnologically. So the main reason for the cancer, it is due to the mutation of the oncogene responsible for the normal cellular behavior. Cellular behavior means that only controls the cell division. Cancer is nothing but uncontrolled cell division only, because the gene responsible for the normal cell division is getting some kinds of defected through the mutation. And this can be detected early stage biotechnologically. Select that particular DNA to be tested to identify the mutation. First, we have to make it a single stranded through denaturation and prepare a complementary strand for that single strand DNA artificially without any sequential defect. Correct sequence only, with the radioactive nucleotide only, and that only we call it as what? The probe. then allow the probe to hybridize with what the single-stranded DNA taken from the patient. So the DNA that we prepared artificially is having the correct sequence. Whenever it is go for hybridization, if it is having that mutation, that area never produce hydrogen bond, so that area is not getting the radioactivity. So it can be cloned, and this defect can be appear on the photographic film. Mutated gene will not appear because that hydrogen bond will not be there with what the probe in that particular area. Right. So this is what the double stranded DNA sample we are taking. Then we have to denature it and convert it into single stranded DNA. And this is what the DNA we are suspecting what this mutation, and this we have to hybridize with what the radioactive DNA probe. We have to make what the complementary strand here. If that complementary strand is having what the real sequence, if any defect is there in this what the strand taken from the patient, whenever it go for hybridization, hydrogen bond will not be produced between these newly constructed artificial strand with the strand taken from the patient. So that will not appear in the photographic plate. This is what the way through which we detect what the defect of the DNA. So this is what the after hybridization. Now this is what the radioactive probe only that only connected with what the DNA taken from the patient. If here it is what matching A, T, G, Z, like it's matching. If it is what the defected here instead of T, suppose it is what the G only, A never connected to the G, right? So that this will not be there. This area will not get what the radioactivity. So whenever we expose to that photographic plate, this never appear because no radiation by the G. This is what the way through which we identify the mutation in the mutation in the DNA. And another one, that is enzyme linked immunosorbent assay only, and it is based on the antigen antibody interaction. So after infecting an individual with a pathogen, naturally the antigen of the pathogen will be there in the infected individual. This can be detected through different means or technology, but the antigen present in the body of the individual is very low in the early stage. And these antigens are type of protein like a glycol protein only, and it is there in the body of the pathogen that will be there in the blood. And early stage, the amount of antigen present in the infected individual is very less, so that can be what you can say detected only through the PCR technique. Through PCR, we can multiply the content and we can observe or we can detect its presence in the early stage. Then come to another use of the biotechnology to produce what the transgenic animals. Transgenic animals. These are animals having a foreign gene. Transgenic animals are the normal animal only, but having a foreign gene, and that gene is also expressing a protein. We are introducing a foreign gene in the body of an animal, and this introduced the foreign gene express a foreign protein in their body, and such a type of animals only called as what the transgenic animal. Maybe heard about transgenic rat, rabbit, pig, sheep, cow, fish, etc. And 90% of the existing transgenic animals are mice only. And common function of the transgenic animals are to study the regulation of gene, to control the normal physiology and development of the individual, to understand the development of the diseases in the individual, to produce biological products to treat a disease, and to test the safety of vaccine, and to test the toxicity of drugs. You maybe heard about the vaccine safety test. After the invention of what this vaccine, we have to test in different level. For example, in case of corona, we developed the vaccine years back, but it came to us only after many months, because there are some procedure to test the safety of this vaccine. Normally, we test the safety of the vaccine in the transgenic animals only. And here you can see a transgenic animal, gene of choice It's a manipulated DNA taken from some other individual, and this we will insert into the egg or taken from what you can say cow. This egg is donated by the cow, and it is what the foreign DNA that we will insert and become the part of its genome. And this in what you can say cell is taken into the uterus of this cow, and egg is implanted. It's a diploid one only, and normally what happened, it go for further development and produce what kid here. And this is showing what a foreign DNA that we introduce from the other individual. This is what the way through which we are creating the transgenic animal. Foreign DNA we insert into the genome of what the diploid cell taken from the body of the individual. Then this diploid one is taken into the uterus and implanted it, and finally go for all kinds of development and give birth to the young one. See, isolation of primary cell, generation of the transgenic cell, and this transgenic cells, we have to take into what the anucleated oocyte. Egg cell only we are taking here. Any cell can be inserted, but it should be what a diploid one. And here we are removing what the nucleus from the egg and making it what anucleated, and keeping what the nucleus of what a transgenic cell, and this can be go for further development. After getting the gastrula stage, this can be implanted on the uterus of the mother, and it is called as what the recipient, and it is giving birth and produce what a transgenic livestock, and it is producing the characteristic feature of what the transgenic cells only. That foreign is not at all producing what characteristic feature, because the gene present in the oocyte is also taken outside, so no chance to produce any characters of this cow, right? And here the transgenic sheep and lamb. You maybe heard about the Dolly. 1996, the Dolly, the sheep was cloned using cell from the 60-year-old sheep udder by somatic cell nuclear transfer. That means nucleus of this cell, it is what the diploid nucleus only taken from what the udder cells, and from the 6-year-old sheep. And this diploid cell, this nucleus transferred into what the egg of an individual and keep it in the uterus. Then go for birth and produce what the first transgenic sheep, and produce from the somatic cell taken from the cells of what the udder region of 6-year-old sheep, through what the somatic cell nuclear transfer, SCNT technology only. Then the 1997, next year, the world first transgenic lamp Polly was also created using the same technique only, contained human gene for the blood clotting factor 9. So taken the gene from the human being responsible for the blood coagulation factor 9, that also introduced into the genome of this lamb Polly, and we prepared in 1997. It is what the transgenic lamb Polly. Right. Then see reason for the production of transgenic animal. For what purpose we have to produce such type of animal? To study the normal physiology and development, to study the different kinds of disease and its origin, to get what the biological product of human interest, and to test the safety of vaccine, or to test the chemical, to test the safety of some chemical. Normal physiology of the individual can be studied by using what the transgenic animal. By introducing gene from the other species, the production of the growth factor can also be regulated or altered. Whenever we are introducing a foreign gene into the body of another species, naturally this gene can disturb the normal functioning of the body in that recipient. That can be studied. The biological effect of the particular gene in an individual can be studied. So that is what the one use of what the transgenic animal. And see, this is what the human placental growth hormone. So we introduce what the human placental growth hormone producing gene into the mice you see the size of the mice. And it is what the normal one, and see the size of the mice. So normal physiology has changed or its work is changed. So that can be studied through what this transgenic individual only. And study of diseases. Many transgenic animals are designed to increase our understanding how gene contribute the development of a disease. So investigation of no treatment for the disease is made possible. Today, transgenic model exists for many human diseases such as cancer, cystic fibrosis, rheumatic arthritis, and Alzheimer's syndromes. Then for getting the biological products of human interest. So some biological products to treat human disease can be prepared biotechnologically. So transgenic animals that produce useful biological product can be created by introducing the portion of DNA or gene. A foreign gene can be introduced in the body of animal so that it produces some biological products. Example, alpha antitrypsin. It is produced by some animal only by keeping a foreign gene inside their body, and it is commonly used to treat the emphysema, a respiratory disorder. In 1997, the first transgenic cow, Rosie, maybe heard about Rosie, produced human protein enriched milk. That is, this milk consists of 2.4 gram per liter. Normally, you know that cow is producing milk for its cubs only, not for the human being. So all the protein present in the milk produced by the cow is for the development of the cub. But we developed what a transgenic cow called Rosie, and it is producing what the human enriched milk only. And this milk containing human alpha lactalbumin and was nutritionally more balanced and product for the human baby than the natural cow milk. Natural cow milk is not at all good for our health, but this milk produced by the Rosie is very much because it's containing what this human what you can say protein only, right? And this is what the Rosie that we developed through this what the DNA recombinant technology, right? And vaccine safety. I told you after the manufacture, after the discovery of a vaccine, there are so many stages to test the safety. Then only we can use it in the body of a human being. And normally we are testing the safety of this vaccine by injecting that developed vaccine in the body of what such type of transgenic animal only, animal having some kinds of what you can say human genome, so that we can easily understand what will happen, what will be the effect of such type of vaccine when we introduce in the body of human being. Right. Then transgenic mice, most widely used model animal to detect gene expression, and used in what knockout technology to detect the gene functions, right? And the chemical safety testing. If you want to know the problems due to the water pollution, how it affect what this life of the aquatic individual. Actually, after introducing that polluted water in the normal water medium, aquatic medium, it takes time to identify the effect of its what in the aquatic environment, particularly the life of the fishes. But it can be detected so early by making this fish so sensitive. How can we make this fish so sensitive? By keeping some kinds of what gene in their genome. After keeping some foreign gene in their genome, so naturally this fish becomes so what you can say sensitive, and we have to introduce what the polluted water. Even the rate of pollution is so slow because of what the sensitivity. It will die. So we can easily identify what will happen due to this pollution after some years. That is what the chemical safety testing. So to know what you can say toxicity and of the water, transgenic animals are made that carry a gene which make them more sensitive to the toxic substance than what the non-transgenic animal. They are then exposed to the toxic substance and affect the studies. Toxicity testing in such animal will allow us to obtain the result in a less period of time. Transgenic fishes are very commonly used to study the pollution of water. If we are keeping ordinary non-transgenic fishes, it takes time to understand the effects. But if you put the transgenic fish, because it is very sensitive, so easily we will get the result. So this is what the way to get the result in the early stage by using what the transgenic animals. So fishes, normally we make it very sensitive by keeping some kinds of this gene inside. So that's all about what you can say, the transgenic animal, then some ethical issues. Of course, we are doing all kinds of manipulation in other individual by introducing the gene, but every individual present in the nature is having its own importance to maintain the ecological balance. This equilibrium should be maintained. Every individual is having its own role in the environment to play to maintain that equilibrium. What will happen if you change the chemical nature of the genetic material? Because nature made such a chemical nature in the genetic material to make them to perform a particular role in the nature, especially to maintain the equilibrium. What will happen if you are making such a changes in the genetic material of the individual? That will question the equilibrium, and that will create a lot of trouble in the balancing of the nature. We should not do it, but it is only for what the human welfare or for the human purpose only. That is what the ethical issues. We have no right. See, we are all just like what the other individual. For nature, we are like other individual only. Right. We have no right to change the chemical nature of the genetic material of any other individual, and it is having their own role in the nature to maintain the balance. That is what the ethical issues. Then come to the patency. And patent means what? After the discovery of something like some machine or something, what you can say, I mean, a chemical produced by the individual, that company or government can go for patency, right? A simple topic only, just to go through it because we have some uh, the time limitation. So go through the patent, and now we have what you can say. These are all what our biomaterial only. See that basmati, then neem. These are all what you can say our own material only. Basmati rice we are using for many years, many centuries, and but you can say neem we are using for what the many years as what medicine, and we are using the tamarind, for many years as what antiseptic material. But now even then it's what our own material, we are not able to use it for any kinds of commercial product because patent is with the American company. They only identify the gene, they only made what the new variety, and they go for the patency. They are getting the patent legally, so that we will not able to make any kinds of product with such material commercially in our country. If we want to create, we have to give the compensation to the company, we have to give the competition to the particular what you can say nation, we have to give compensation for the scientist also, then only we are permitted to make any kinds of product with such material. If we are not making such kind of material, if we are not getting what the permission from the country to make the material commercially, and this is a serious issue only, that only we call it as what the biopiracy. Biopiracy. Okay, it is used for the utilization. This term is used for the utilization of the bioresource by multinational companies and other organizations without proper authorization from the concerned country and people concerned, without the compensatory payment. Most of the industrial nations are financially and technically rich but poor in biodiversity. You see, biodiversity is rich in what the underdeveloped countries and developing countries, but these developed countries are simply exploiting the underdeveloped and developed countries and getting their bioresource, and they are earning a lot of money through the patency. The underdeveloped and developing countries are rich in biodiversity and traditional knowledge. So developed countries are exploiting the traditional knowledge and bioresource of underdeveloped and developing countries without giving proper compensation. And some countries are developing law to prevent such unauthorized exploitation of their bioresources and traditional knowledge. And we, India also, what you can say, passed many laws very recently, what you can say, to avoid the misuse of our bioresource and traditional knowledge. And these are the laws. Indian Parliament has recently cleared the second amendment of the Indian Patent Bill to see the issue related to the patent, particularly patent terms, emergency provision, research, and developmental initiatives. So that's all about what this chapter. And if time permit, I will discuss a small question because it's asked by a person, and take only 5 minutes to do it. Just 1 minute. Change to that question. I will discuss that question.
Okay, sir.
That question, no? Then another slide, I kept it there. No, not that one. Just close it.
Okay.
Then
Another video.
that PDF, not PDF. It is PowerPoint only. So name given as question. It is there in the desktop, I think.
This one is it?
Question.
This
Right. Yeah. It is on love now.
Yes.
Right. So all of you listen. I won't take much of your time. So this is what the last section. That's why I am going to discuss this question. Yesterday, one guy contacted me and asked to, right, 2 minutes, right, asked to solve this problem. And this same question asked in the first time examination also. Consider a population of 100 cats, of which 84 are black cats and what you can say 16 are white. Assuming that the cat color is a single gene inherent. That means what? It is monohybrid cross. So calculate the number of heterozygous cat, and these are the options given: 36, 40, 48, and the 84. Then how to do it? Here we have to use that equilibrium, Hardy-Weinberg equilibrium. We have to use it. In Hardy-Weinberg equilibrium, all of you know that is what the p square plus 2pq is plus q square is equal to 1. This is what the equation. P square plus 2pq plus q square is equal to 1. It is just like what our monohybrid genotypic ratio. Geno, not phenotypic. And see Mendelian experiment that tall plant, homozygous tall plant cross with what the homozygous recessive, and it produce what the gene capital T small t go for cross and produce what capital T small t, then it's go for second generation, and here it produce two type of gamete, capital T then small t. It also produce capital T small t. You may be getting it. These are the gamete produced by one parent, and this is what the gamete produced by another parent. And go for crossing like this. This is what the male gamete, produced by the male individual with the capital T and small t, and this is what the female gamete or gamete produced by the female. This is what the male gamete, and these are the female gamete. Here it will be capital T, and this is what the capital T, small t, and this will be again capital T, small t, and this is what the small t, and here phenotypic ratio is 3:1. All of you remembering that phenotypic ratio, one tall and 3 tall and 1 dwarf. That is phenotypic ratio. But genotypic ratio is what? 1:2:1. Two homozygous dominant. One homozygous dominant with the capital T, heterozygous 2 numbers, and homozygous recessive only 1. And that only mentioned here p square plus 2pq plus q square. P square means what? This is what the p square, homozygous dominant. And the q square only 1, that is what? Small t only. And 2pq, that is what? Capital T, small t. Here 2 capital T, small t, that is what? The p square only. And if you take the population, that frequency is always constant, that is 1 only. If it is changed that equilibrium, naturally chances are there to appear a new character. That only lead to the evolution. Only 2 character here. Either this will be a tall or it will be a dwarf. So its frequency is, total frequency is always 1 only. P square plus 2pq plus q square is equal to 1 only. So this is what the idea. Then how we will use this idea to solve this problem? See this problem, how to solve it. And here the recessive one total number is 16. That means what? The p square is 16. P square, q square is what? The recessive homozygous recessive, it is always a 16. And out of 16, out of 100 it is 16, means out of 1 we have to take, because p square plus 2pq plus q square is equal to 1. So we have to take it in 1 only. So divided it with 100. So we will get what? The 4, right? Q square is 4. Here it is q square, it is what? The 16. Out of 16, this will be 4 only. 1 minute. Have a doubts here, right? I will solve it and I will show you here. This is what the solution I solved here. This is what the p square plus 2pq plus q square, that is what? 1 only. And the total number of recessive allele, that's q square, he found out of 100 only 16. So the q square is what? 1.16. Out of 100 it is 16. So in 1 it is only what? 0.16. And is q square, it is what? 0.16. But q is what? The 0.4 only, because it is what? The square root of what? 0.16. And another one that we already got the value of the q. We got the value of the q here, and the q is what? 0.4 only. Then we have to get the value of the p. How to get the value of the p? Then p plus q is equal to 1. P plus q is equal to 1. We know the value of the q, that is what? The 0.4 only. And to get the value of the p, we have to minus 0.4 from the 1. That is what? The 0.6 only. So here p is equal to 0.6 only. P is 1 minus 0.4. That is what? The 0.6 only. Then we have to go for solve this equation. P square into 2pq. P square is equal to what? 0.6 into what? 0.6. That is what? 3.36 only. And here we have to find out the q square. And q square is what? 0.4 into 4 into 4. 0.4 into what? The 0.4, that is what? 0.16 only. 0.16 only. Then we have to find out what? The 2pq. And this is what? The total number of heterozygous condition. It is what? The homozygous dominant. It is what? The homozygous recessive. the total number of what the homozygous dominant is 0.36 out of 1, and homozygous recessive is what the 0.16 out of 1. Then 2 PQ value we have to find out. So 2 PQ is equal to 1 minus, see 1 minus, we know the value of P square and Q square. Then we have to find out the P2 PQ. From 1, we have to minus the value of P square and Q square. So that, so here 2 PQ is equal to 1 minus 0.36 plus 0.16. So 2 PQ is what? Minus 1 minus 0.52 only. Add these two and get what? My 0.52 only. So 2 PQ is equal to what? 0.48. And out of 100, it is what in one only here, what you can say, P square is equal to what? P square plus Q square plus 2PQ is equal to 1. That is what the formula here. And here P square is what? The 0.36, and Q square is what? 1.16, and this 2PQ is here. You can find the 2PQ is what? 0.48, and get what? The 1. And out of 100, it is only out of 1. Out of 100, this will be what? The 48. 0.48. 0.48 into 100. That is what? The 4.8 only. So see the correct answer here, right? See the correct answer. Consider the population of 100 cats, of which 84 black and cats and 16 are white. Assuming that the coat color is what? The what you can say, single gene inheritance only, and calculate the number of heterozygous. That is, heterozygous is what? The 2PQ only. You have to find out the 2PQ. It is what? The 48 only. This is what the correct answer. So this is what the way to find out the answer for this question. And here you have to use only the Hardy-Weinberg principle. So with this, I would like to wind up this 4 days section. I think actually I came here for all kinds of preparation. Because of the time limitation, I was not able to, what you can say, perform well according to your desire. Of course, some of the things are useful. Collect a maximum question from the different sources and discuss with your teacher, discuss with your friends, and utilize your time properly, and perform well in your examination. And biology, learning biology is not only for getting good mark in the examination, because biology is what? The study of life only. So that you should learn how to live practically. So that also you have to take care. And for any kinds of doubt, you can contact me. And my name is what? Minil, MINIL. And you can contact me in the number. So my number is 9496636167, but not in the odd time, not in the academic time, right? Anytime you can call other than the odd time and academic time. So any doubts, we will clear it, and try hard, and try to come up in your life with the expectation of your teacher, school, and your parent. And of course, if you work hard, you will able to come up with the flying colors in your life. So all the best, and God bless you. Thank you so much.